very brief introduction of our institute. We are very close by, 5 minutes away from here, on Splaiul Independenței. The Institute of Biochemistry belongs to the Romanian Academy, and since 2012 we are also hosting the Proteomics Center. Then with European funds we have developed several facilities that allow us to perform advanced techniques such as mass spectrometry, also confocal microscopy, enhanced bioinformatics and computing techniques, and also quantitative histology. We are 5 departments, and we are actively collaborating with different centers all around the world. In my department of molecular cellular biology, we are mainly focused on studying protein traffic and degradation using model cell lines as well as C. elegans, and through collaborations we are also performing in vivo assays. My focus after I have done my postdoc at Northwestern University has been to study 2-transglutaminase, which is a very sticky protein that favors cancer dissemination and metastasis in various solid cancers. We are testing small molecule inhibitors for this protein, and also identifying drug escape pathways that we try to co-target. And also I identify myself as a flow cytometrist. I am developing and using assays, multicolor assay, to characterize the tumor microenvironment. And together with our team in the Institute of Biochemistry, and under the coordination of Dr. Livia Sima and the 2 teams in FLPR, we have recently started also to study flash effects on cancer cells, as well as relevant immune cells in the tumor microenvironment, and trying to decipher how the cells are communicating in the context of irradiation-induced stress. Also, I am maintaining active collaboration with Dr. Daniela Matei, who recently moved to Houston, Texas. And she is an oncologist studying ovarian cancer, and a very good advisor to keep our research relevant. So the oncologist, when a patient presents with a tumor, has several tools available, among which radiation therapy has been a key component since almost 100 years ago. But as also Dr. Radu explained, this ionizing radiation, this conventional ionizing radiation, produces, along with the anti-tumor effect, many unwanted side effects. And therefore, the flash represents a true alternative that has to be dissected, understood, and applied for therapeutic treatment of cancer. At the cellular level, when we apply X-rays, depending on the dose applied, there are several processes that are occurring, such as DNA damage, gross accumulation, lipid peroxidation, and following these processes, the cells are either incapable of mitosis, or they are surviving, they are repairing their DNA, but they show reduced clonogenicity. A very recent review in New England Journal of Medicine has shown the very dramatic effects that occur long-term after the treatment of patients with conventional radiotherapy. The normal tissues are affected, fibrosis occurs, vascular injury, and also secondary cancers. So the motivation for our work on flash was to contribute to the development of a new laser-induced very high-energy electron flash irradiation for radiotherapeutic application using the 1 petawatt high-power laser facility at CETAR in FLPR, and also to understand the molecular mechanisms of cancer and normal cell differential response to these ultra-high dose rate irradiation regimes. I also want to mention this paper by Pavodon, which opened again the flash research field, and who studied lung cancer in mouse models and also received a Lifetime Achievement Award in 2025 for his contribution to this research. He very nicely showed that even at high doses, 20 gray doses of flash, the mice that are exposed to this type of radiotherapy are surviving, as shown by these Kaplan-Meier plots. And also, the fibrosis in the lungs is not visible when the mice are treated with this alternative flash irradiation. So this radiotherapeutic regime could represent a new cancer treatment modality, especially for deep-seated tumors that are very hard to treat with other types of tools and radiation. So they went on and did a first flash clinical trial on cats. So as you can observe here, after applying flash radiotherapy on squamous cell carcinoma of the nasal planum in cats, after 14 months the tissue is healed, and they observed the protective effect of using this radiation in this animal model. And then they received a patient who was multi-resistant in his T-cell cutaneous lymphoma, so he was several times irradiated with conventional radiotherapy and nothing worked. And they successfully treated his skin cancer with this therapy, and they developed also a flash knife that is used to apply electrons that are accelerated via LINAC. So it's a particle accelerator strategy. Then on ovarian cancer, there is the group of Billy Liu in Stanford, showing here on a non-tumor bearing mice that applying the same dose of flash as in conventional therapy, the survival of mice is almost 100%, while the others that are treated with conventional radiotherapy die in 10 days post-irradiation. Also here they show a bouncing back of the body weight, and at the gut level, a regeneration of the gut epithelia within 12 weeks post-irradiation. That does not happen as efficiently with conventional irradiation. So what we look at when we want to study molecularly the effects of radiation on cells, we look at these key players. First, we look at DNA damage by looking at gamma H2S accumulation at the DNA damage points. And also we look at ROS accumulation and the proteins that are involved in cell signaling in response to radiation. The gamma H2S is recruited to the double DNA strands produced by radiation in the nucleus. And this phosphorylated histone has a peak of accumulation at 30 minutes post-irradiation, and then following by immunofluorescence microscopy these effects at later time points, we can see if the cells are able to repair their DNA, and we observe less foci, or they maintain or accumulate further DNA damage. So we did a first experiment in Chetal using the 1 petawatt high power laser, which by heating gas jet produces a mixture of radiation that is very rich in electrons. And first we decided to look at a co-culture of melanoma and normal melanocytes that we seeded in a slide flask, and we monitored the irradiation dose by 3 different ways, using thermoluminescent dosimeters that are intercalated between the flask cells, the chromic film that was attached on the bottom of the flask, and also real-time monitoring in the ionization chamber. We published the results in scientific reports. I just briefly want to show you our strategy to look at gamma H2S foci. So we have scanned using our automatic scanning microscope. We have scanned the slide upon labeling the cells with antibodies against gamma H2X. In parallel, we had the scanning of the chromic film that was developed, and you can observe here the higher doses in the middle of the slide and a gradient of dose distribution along the slide with cells. And we have tried to correlate different regions exposed to different doses with the effect at the level of DNA damage. For this, we have segmented the cells, and based on elongation of nuclei, we were able to differentiate between melanocytes, which have an elongated nuclei, and melanoma cells, which have round nuclei. So by segmenting these two subpopulations, we could look at the differential effects in the two cell types. We always use cisplatin as a positive control of DNA damage inducing factor. And here are the psychometric strategy that we use that is very similar to the one used in flow cytometry. So the result was that for one of the slides, for the top area in the slide, we observed significant differential response between increased DNA damage in melanoma cells and no effect on melanocytes as compared to the non-irradiated control. We next published this method in a bioprotocol, and we also were invited to record a webinar on the tissue gnostics platform. So, but the tumor microenvironment does not contain only cancer cells. It is a very complex environment which has several cell types, including immune cells, fibroblasts, cells in the blood vessels. So there are a lot of things that are unknown that happen when we apply radiation. This is just to show you that there are methods, multiplex immunohistochemistry methods, that allows us to visualize multiple subpopulations of cells, immune cells, and cancer cells in different type of tumors. So the microenvironment is very dense and very complex. So several questions have remained. What is the cell fate after the flash radiation using petawatt laser-induced electrons? Is the DNA damage repaired? What is the molecular mechanism responsible for the differential response? How does different cell types respond to treatment? Is there a benefit to combine this therapy with the immune checkpoint inhibitor? So this motivated us to start a new project. We have a new grant that we are working on in collaboration to increase the understanding of molecular mechanism involved in response to flash irradiation. As models, we use two cell lines. One is Menjusol, which is a melanoma cell line that is representative for the early stage tumor progression in melanoma, and SCOV3 cell line is an ovarian cancer cell that is the most resistant, the most chemo-resistant cells to standard of care. So we do, with these two cell line models, we started to work, and we have planned to use many assays and strategies to understand the effect of radiation on this type of cancers. So the gold standard is to perform the clonogenicity assay, to look at survival fraction after irradiation. We also perform gamma H2AX labeling to validate the DNA damage effect of the dosage applied. We look at two model proteins that are important in melanoma and ovarian cancer that are upregulated and important for resistance to chemotherapeutic and radio treatment. And also we are planning to work this year on immune cells to look how the media secreted by the cells that are irradiated are activating or modulating the immune activity of these cells. Alexandra will present later on also scaffold models that we can use to test cell invasivity for our project. So first, we have started, we went back to conventional X-ray, but adding an innovative new pulsed mode of applying radiation to cells. So using a chopper that is rotating in front of the X-ray source, we can produce pulses of radiation that are applied to cells that are vertically hold in front of the X-ray source. So we use the slide flask, and we seed cells. We fill the flask with media, and we apply the treatment. That takes a few minutes. And then the strategy is to recover the cells in the incubator for 30 minutes, and then harvest the cells for further analysis by immunofluorescence microscopy to look at DNA damage, expression of TG2, expression of proliferation markers, accumulation of ROS by flow cytometry, and the other time point is 24 hours, where we want to see if there is any potential DNA repair after irradiation, and look at cell motility during the early stages of recovery after irradiation, and are also detaching the cells and seeding them in a clonogenic density to look at the clonogenicity potential. We always use cisplatin as control throughout our studies. As early as 24 hours, we already can see a dose-dependent modification of the appearance of the cells. The nuclei are larger. There are a few cells that are detaching and entering apoptosis. And next, we have tested the different cell seeding densities of the cell suspension we obtain after irradiating with different doses, and you can observe a dose-dependent effect of decreasing the clonogenicity in these cells. The survival fraction for SCOV3 was 0.1%, while for Medusa was 1%. They are more resistant to radiation. And interestingly, we kept the cells for 3 weeks in culture after irradiation, and we performed again clonogenicity assay, and we observed that for doses higher than 2 gray, there is a very low cell expansion in clonogenic density for these doses, while the 1 gray makes them stronger. So it's very important to choose the dose and to see the persistent effects on longer time points. Then we have started to look at pulse radiation versus conventional, and we quantified gamma H2AX mean fluorescence intensity in cells that were subjected to 3 doses of pulsed X-ray versus 3 doses of conventional X-ray that are highlighted here. We also, as a control, added cisplatin to each of these doses just to make sure that we observe an enhancement of the DNA damage in blue bars. And what we interestingly saw is that for 2 gray, we observed an increase in DNA damage if we apply this dose using the pulsed setup versus the conventional setup. To show very shortly why we use TG2 as a marker of response to radiation, we have also a TG2 knockout cell line that I obtained during my postdoc via CRISPR-Cas9 genome editing, and one can see here an increase in the DNA damage produced by cisplatinum or a higher dose of radiation when the cells do not express TG2. And when we perform metabolic assays, we saw that for the TG2 knockout cells that were subjected to 4-grade radiation, the metabolic rate of glucose is decreasing. So the cells are more sensitive to DNA damage if they don't express TG2. Also, when applying 4-grade radiation, we can consistently see a decrease in clonogenicity. And also, when we compare either TG2 knockout or irradiation with 4-grade, we observe a decrease in the expression of stemness markers that are known to be involved in the stemness of ovarian cancer cells, like ALDH1A1 activity.
2 minutes, sorry, 2 minutes.
2 minutes, okay. So I will pass very quickly. We look at TG2 expression. We saw that there is a difference between conventional and pulsed irradiation, while in conventional radiation, TG2 expresses higher with the higher doses applied. For pulsed irradiation, the expression grows after 6 days. There is no significant difference in Ki-67 expression, a proliferation marker, except the one that for 2 grays there is a slight decrease in the expression of this nuclear marker, which is consistent with our holomonitor data, which shows that within the first 24 hours of irradiation, there is a very small decrease in proliferation of these cells. We observed an increased apoptosis, a dose-dependent increase in apoptosis in both cell lines, which is not visible anymore after 3 weeks of cell culture. And then the second approach was to irradiate cells in suspension in order to be able to perform chromogenicity assay due to the very small area that can be irradiated. We reconfirmed DNA damage, and we saw for the 2-gray pulse setup a decrease in chromogenicity versus the conventional mode. Performing cell cycle, we saw a dose-dependent increase in DNA content in the Melchusov cell line, while the SCOV3 does not show a very clear response, but using cisplatinum we can observe that our assay is consistent and we are able to detect increasing DNA content when it exists. Very interestingly, by counting the number of cell division per cell for each type of dose and cell line, we can observe a dose-dependent increase in the cells that are dividing less when we apply a higher dose. And it's very interesting to see that for both cell lines, 2-gray pulse irradiation produces less number of cells, less divisions than 2-gray applied conventionally. We also did a motility assay, and Alexandra will show this later on. We observed a decrease in motility in both cell lines, and also further accumulation of ROS in SCOV3, while the Melchusov are able to partially compensate for ROS and showing antioxidant activity in a subpopulation of cells. We then performed a workflow to integrate C-force analysis for measuring cell respiration by measuring oxygen respiration rate, and by this we were able to show distinct responses function of the dose applied. For the highest doses, there is a less efficient coupling of the oxygen consumption and ATP production, and also a higher stress and metabolic rate in the cells that were treated with cisplatinum or with the highest dose of radiation. Very quickly, the last experiment was to try X-ray flash in a new facility that was developed recently in Inefere. So they applied in several points of the slide class different doses of X-ray, and we were able to see in co-cultures of ovarian cancer and melanoma increase in DNA damage with the dose that is 10 times higher than the other one. So with increasing the dose 10 times, we can observe DNA damage. We can also see for the first time micronuclei, which are proof of a very high impact of the radiation of the exposed cells. And here is the melanoma cell line that is very able to repair its DNA 24 hours after exposure to 2.7 rays of flash X-ray. So next we are planning this year to correlate and integrate our transcriptome data with secretome data by mass spectrometry, and also to perform immune cell profiling by C-course analysis as well as immune assays, and to try to integrate this data to understand better the differences in cell response to different radiation regimes. And this is just to show the amount of work and sometimes danger that we expose ourselves to work on this project, and that is very satisfying and very rewarding for the entire team. Thank you.
Thank you very much. A lot of very nice results. I'm sure you will enjoy many questions during the lunchtime. For now, we can only take one. So, anybody has a short question for Dr. Livia Sima? If not, I will ask one. So, in this very interesting field that you show, what is the next big breakthrough that you expect in the next 5 years? What do you think that it's almost there to happen, but not yet? So,
related to flash?
Yes.
Related to flash, if we talk about the laser-induced flash, it would be to really control it and to be able to apply controlled doses and also discover the key cell death mechanism as a response to this type of radiation. So to really know exactly how to apply it in a controlled manner and the exact mechanism by which the cells die and the doses that is necessary really to only affect the cancer cells.
And do you think this is, let's say, close of achieving, let's say, next 5 years, next 10 years?
Maybe. There are a lot of efforts and funding that is invested in this direction, so we are hopeful to have these results.
Let's hope.
Yeah.
Thank you very
Thank you
much.
so much. Thank you.
Thank you once again.